Good morning to Dr. Muhammad Reza Rahman, our lecturer for course KNC 2133 Analytical Chemistry. So today we are going to present our topic, which is differential scanning calorimetry, DSC. So we are from group 7. My name is Muhammad Hafizul Hazmi bin Hossein, and I'm going to present introduction and conclusion. Next will be
My name is Muhammad Fadila Aiman bin Sulaihi, and I will be presenting the description and theory.
My name is Muhammad Khalif and I'm going to present methodology.
My name is Faizul and I will be presenting about result and discussion.
So let's start our presentation for today. So introduction, we have a background about DSC. So basically, historically, ceramic industry are the first one to study about the temperature transition back in the 1800 by using the differential data analysis DTA. This method is done by placing the thermometer into the material then heating inside the oven. But there are serious problems happen due to the placement of the thermometer, which is not reproducible. Then, in the year of 1960s, Mike O'Neill uh, come up with a solution, which is in order to measure the heat flow, the movement of heat in and out of sample, he produced a new uh, heat flow, which is called DSC. So next will be the application of DSC. There are many applications of DSC in our industry. They are such as crystallization time and temperature, heat of melting and crystallization, percent crystallinity, oxidative stability, computational analysis, heat capacity, heat of cure, and many more. Next will be the advantage of DSC. One of the benefits of DSC is that it has become, because of it relies on the heat measurement, the use of differential scanning calorimetry in measuring biomolecular stability without actually requiring optical transparent sample can be defined by native biomolecule. The next one will be EDSC is the simplest and fastest method of calorimetry that can be used in the laboratory. And last but not least is when it is used uh, in studying of the mineral oil, the moisture within the oil does not influence the effect of DSC towards the value of the experiment. And next will be the disadvantages of DSC. DSC is a very highly costly and not commercially available for us. And next, it is less feasible because its temperature range performance is smaller compared to DSC flux. The next slide will be the limitation of DSC. DSC is now a routine technique and the methods are relatively inexpensive, so it can be used various laboratories for characterization. It's enabled the cost-effective determination with moderate effort. DSC the demonstrate other drawback. This is meant by uh, it has a good in speed direct approach to assess a thermal stability, but the concentration limit can cause the difficulty when the quantity is operated in larger size of a value. And next will be the application of DSC in milk product restrict the sample to a limited quantity or size to be evaluated. The small sample size is only suitable for one hand, which means less analytical material that can restrict the amount of data when performing the experiment to obtain average value. And last is the analytical equipment. So we have two, which is DAC 8000 for power consumption and DAC 4000 for heat flux. So next, I will pass it to Muhammad Fadilah to present.
Thank you, Hafizul. So next, I will present the principle of differential scanning calorimetry in description and theory. So differential scanning calorimetry, or DSC, it is a thermoanalytical technique used to measure the temperature and heat flows in terms of materials transition. So this technique is used to measure the enthalpy changes due to the changes in the physical and chemical properties of a material as a function of temperature or time. So when a transition occurs during the process of heating of a sample, it means that there is a slight difference in temperature between the sample and the references. Sorry, and the reference shows next. So DSC also measures the energy absorbed or released by a sample as it is heated or cooled. According to instrument specialists incorporated, the basic principle underlying this analysis technique is that when the sample undergoes transformation, such as phase transition, more or less heat will need to flow to it or need to flow to the sample than the reference to maintain both at the same temperature. There are two types of differential scanning calorimetry. The first one is heat flux PSC. This, fact, this heat flux PSC belongs to the class of heat exchanging calorimeters. Uh, the primary measurement signal for this type of DSC is the temperature difference, whereby it determines the intensity of heat exchange and the resulting heat flow rate, which is proportional to it. So it measures the temperature difference, which is Ts minus Tr, whereby Ts is the temperature of outside surface of sample holder and Tr is the temperature of reference holder. So this is the block diagram of heat flux. Heat from the heater is applied to both sample and reference through the heat sink and heat resistor. The changes in heat sinks and holders are related and interconnected with the heat flow, where the heat flow is proportional to the heat difference of heat sink and holders. Compared to the sample, heat sink has enough and sufficient heat capacity. So when a when a transformation or when a transition or reaction occurs, the heatsink will act by compensating the heat from the endothermic or exothermic reaction. Thus, the temperature difference between the sample and the reference is kept constant. In the sample and reference, the difference in amount of heat supply is relative to the temperature difference of both sample and reference holder. The quantitative measurement of unknown sample can be done by calibrating the standard material. So this is the schematic diagram of heat flux PSC. So the other type or another type of differential scanning parametry is called as power compensation DSC. This type of DSC utilizes the thermal energy difference between both sample and reference per unit time. The purpose of power compensation DSC is to measure the enthalpy change of sample holder with the consideration of time function simultaneously. During heating up of sample, the same heating power is supplied to both furnace or pans via a control control circuit. This step is required because it is required in order to change their mean temperature in accordance with the preset heating. The temperature of both micro furnace or pans is always equivalent when there is ideal thermal symmetry. So, when an asymmetry occurs, a sam- will occur when there is a sample reaction with which result in temperature difference result between the micro furnace or pans containing both the sample and reference. Both the measurement signal and the input signal of a second control circuit are the temperature difference. The second circuit offsets most of the reaction heat flow rate by increasing or decreasing an extra heating power of the sample furnace. So I think that's all for me. Next, I will pass it to Khalif.
Thank you, Muhammad Fadilah Haiman. So I'm going to start with methodology part, which is first thing first, eye protection. So make sure we wear the safety glasses and protective clothing, laboratory coat, and neutral gloves. So let me start from sample preparation that has been done in the procedure. So the first step is the scale was there, a T0 hermetic lid and a T0 pen were placed on the balance. And third is the balance door was closed and wait until the balance stabilized before recording the weight. One drop approximately 3 to 6 milligram of each sample was pipette in the aluminum pen using a disposable pressure pipette. Using tweezers, the pen was placed in the blue T0 hermetic lower die and put the die into the T0. The lid was placed on the top of the pen by using forceps. The blue T0 hermetic upper die in the hole was placed above the lower die. To know if it was secure, make sure the upper die could spin in place and not move up or down. The handle of the T0 press was pressed down to seal the pen and lid together. The seal the SC pen was weighed. The weight of the original pen and lid were subtracted in order to accurately weigh the sample. The seal DSC was placed in the desired pen number in the DSC Q2000 auto sampler. So let us move to instrument operation, startup. So the first step is the main power was switched on and wait for the system to boot. The computer of the machine was switched on. The software was open, TA Universal Analysis on the desktop of the computer. The color was started by pressing on in the control tab. When scroll down to event, go to standby, temp was pressed. This was to make sure that the cell did not freeze. And then the nitrogen gas tank was turned on, which would be held at 20 psi, and the sample purge flow should be 50 milliliter per minute. And next is sample loading. The first step is the DSC was opened by pressing the lid button on the touch screen. Second, forceps was used to carefully place the sample pen and empty reference pen on the two pedestals. The empty reference pen should be placed on the rear pedestal and the sample pen should be placed on the front pedestal. The third step is the lid button on the touch screen was pressed to close the machine. Next, let we move to setting parameters. So the first step is by using the acquisition software, Sample information was entered. The concentration were entered if available. Otherwise, enter concentration value into analysis software prior to data analysis. So second step is the option that ensure cleaning of cells with detergent before every sample scan was selected, which should be followed by multiple water rinse steps to ensure no detergent residue was left in the cells. And then the starting temperature of the experiment was set to 20 degrees Celsius, set the final temperature of the experiment to 100 degrees Celsius, then set the scan rate of the experiment to 60 degrees Celsius per hour, which was the common scan rate. And then the samples were rescanned to investigate the reversibility of the thermal unfolding. And next, the unfolding of a protein was considered reversible if the enthalpy obtained for the second scan is at least 80% of the enthalpy value for the first scan. And then the post-experiment thermostat was set to 10 degrees Celsius to preserve the integrity of the calorimeter cells. And then the experiment setup parameters was made sure to be correct before executing the experiment. Next, let we move to running the test. So the first step is to run the test. The green play button arrow on the toolbar was clicked and wait for the data to appear on the graph. And next is, let me move to how to clean up. So the first step is the sample was removed after the furnace and stage assemblies have cooled sufficiently. And second is the reference pan was removed once the testing was done. And the next is how to how the hardware shut down. So the first step is the nitrogen purge gas was turned off by closing the main cylinder valve, and then the computer was shut down, and then the main power switch on the instrument control panel was turned off. Uh, I think that's all for me. Let me pass to Faisal to presenting the result and discussion. Thank you.
Okay, thank you, Khalif. So I will be presenting about result and discussion. So next, so first, the raw data collected from the DSC experiment. The difference in the concentration of the sample used had an effect on changes in the heat capacity recorded by the calorimeter. Additional thermodynamic domains that might include one or more structural protein domains that do not contribute to the transition. A lower concentration may also be revealed at higher concentration. So these figures show the raw data for the DSC experiment that were based on different batch of two protein. So each line in this graph represent a production lot. Protein 1 has three structural domains that are cooperatively melting in the post-transition region of the thermogram. In graph B, protein 2 appears to accumulate more readily upon heating, resulting in an increase in heat capacity above 100 degrees Celsius. Next. Okay, next is analyze the DSC data. From raw data to protein were analyzed, which is protein 1 and also protein 2 that being present in the last graph. So this graph shows the result of the baseline subtraction and iterative curve fitting. After baseline subtraction, the blue line reflects the thermogram, while the red line represents the curve that best match the thermogram. So we can see in this picture, we can obtain the CP or the TM, the transition temperature, and also calorimetry enthalpy. So next, so this is for protein too. Next. So the next one is I'll be talking about incomplete thermogram. Setting the proper temperature range is important when evaluating a non-sample. Uh, Otherwise, as illustrated in this picture, incomplete thermogram could result. Although it is possible to derive the transition temperature from such profile, it is not possible to accurately evaluate the calorimetry heat. The sample must also be retested with a broader temperature range in order to fully capture the thermal transition. So this figure shows an complete thermogram at an insufficient temperature range. In contrast to the experiment from for the graph before this, which was set at 120 degrees Celsius, this graph only showed the final temperature of the experiment was set at 19 degrees Celsius, which did not match the entire transition profile of the protein. Next. Okay. Effect of chemical detoxification on the tertiary structure of protein 1. So one of the most essential physical properties of protein and protein-based product in the industry is thermal stability. It is used in pharmaceutical to assess the safety of biologic under different conditions, including buffering formulation and natural factors such as humidity and also temperature. So this figure shows the analyzed data showing the impact of chemical detoxification on protein 1 tertiary structure. It can be assumed that the toxification step introduced some form of variation in protein 1 structural conformation, which give it this detoxify from greater stability. Next. So this one shows the effect of storage condition on the conformation of protein 3. So this figure illustrates the degradation of a domain which was confirmed by a decrease in molecular weight when the sample were analyzed using size occlusion chromatography with multi-angle light scattering. Based on this figure, the effect of storage temperature 2 to 8 degrees Celsius on the stability and tertiary structure of protein 3 over 30 weeks is shown in this graph. So using DSC method has been successfully integrated into different research packages for characterization, including stability and product comparability tests. The transition temperature of a protein in different buffers and at different concentration can be used to determine the formulation that proffers the most stability to the protein and become an important analytical tool for product formulation studies. It is important to ensure testing parameters consistent from run to run within the same study to ensure the reliability of this method and the objectivity of its performance. However, to accommodate variation in the physical property of different protein, the technique may be changed. So that's all for my part. I will pass it to my uh, group member Hafizul to conclude this presentation.
Thank you, Muhammad Faizul. So, in conclusion, the main objective of the experiment was to determine the thermal stability and conformation of the protein antigen, which was successfully accomplished by the particular methodology used in the experiment. In terms of thermal stability, which is the limiting factor in the most operation, the dynamic behavior, the system versatility, which is temperature and concentration profile, reaction, kinetic, and heat transfer to the system, are the key variables that need to be reconsidered. In order to achieve the objective of the experiment, buffer scanning, raw DSC data collection, DSC data analysis, and incomplete thermogram were found and described in several stages to examine the effect chemical detoxification on the tertiary structure of protein and the effect of the storage condition on the conformation protein. So, in addition, the purpose of the thermogram is to present the outcome of the analysis in comparable heat capacity versus temperature graph, the effect of chemical detoxification on the tertiary structure of protein, and the effect of storage condition on the conformation of protein. And last but not least is the results show that the performance characteristic needed for the measurement of the thermal transition of monoclonal antibodies and various vaccine antigen protein in solution were adequate for the particular use. The result obtained showed the apparent midpoint thermal transition values obtained from the same protein for the same protein, and the difference instrument and analysis are very reproducible, and the typical profile value obtained from the same protein of the same instrument are also strong. DSC, this I can conclude that DSC is an effective tool to identify changes causes by various factors in individual protein and protein fraction. Thank you so much. That's all from us.